I would like to thank the Center for Consciousness Studies and California Institute for Human Science for inviting me to present my work. To briefly introduce myself, I am Dr. Susan Gordon, a core adjunct professor of psychology at National University in La Jolla and research director and psychotherapist at the Southbury Clinic for Traditional Medicines in Connecticut. I will be discussing my newly released second book published with Springer Nature, The Mind-Brain Continuum, Psychoneurointracrinology, and how it impacts the study of consciousness. Drawing on my interdisciplinary background in the history and philosophy of psychology, neurophenomenology, and medicine, this book advances a holistic theory on the development of self, given the notion that intracrinology, the intracellular biosynthesis of steroids, is autopoietic. Autopoiesis, or self-production, derives from the complex interplay between multifactorial influences that are constantly changing and intersubjective. Self and not self are defined by the perpetual interaction among these two spaces. Hormone biochemistry is one of the most fascinating, however elusive, areas of medical science. It is not well understood, does not lend itself to linear analysis, and quickly becomes a system of projection and convolution. As it is impossible to hold one part of the person constant to observe another, we need to speak about hormonal analysis theoretically. I will give you an overview in this presentation and encourage you to go back and read what I provided because there's really insufficient time for this presentation. The Greek word autopoiesis combines auto or self with poiesis, meaning creation or production. In the 1970s, Umberto Maturana and Francisco Varela adapted principles from cybernetics and dynamic systems theory to formulate their theory of autopoiesis, in which they argued that living systems are endogenously controlled, self-organizing, emergent, autonomous, operationally closed, membrane-bound reaction networks. For example, the nervous system is not an input-output information processing system, but an operationally closed network whose elements are invariant patterns of activity in neuronal ensembles that function to create meaning. Living beings as autonomous agents actively generate and maintain their identities and enact or bring forth their own cognitive domains. Knowledge is itself enacted. The next two slides discuss applications of the theory of autopoiesis. In Ecology of the Brain, the phenomenology and biology of the embodied mind, Thomas Fuchs argues that the brain alone is not constitutive for subjective experience. Instead, he argues the brain is a relational organ, not creator of the mind and the experienced world, but a mediating organ that structures the mind itself. Structures of the mind as a plastic system of open loops form from the integrated activity of the living being and are imprinted onto the brain. In this way, mind and brain form a reciprocal relationship between process and structure embedded in the relation between organism and environment. Homeostatic cycles between the brain and body form the basis of pre-reflective self-awareness, basal state, or the feeling of being alive, which through interoceptive signals serving homeostasis are integrated by centers in the brainstem, diencephalon, and hypothalamus. Circularity characterizes the structural dynamics of the organism, part to whole, and the processes of development over time. Between higher and lower level processes, the whole and its parts, we see macro and micro level reciprocal relations of self-organizing systems. A living being reproduces its components, which regenerate the system as a whole, downward, whole to part, upward, part to whole, higher to lower, and lower to higher. Learning and development have a spiral nature. Conscious experience, feelings, emotions, and affect serves the biological homeostasis of the organism as a whole. That is the function of consciousness. Sensory motor cycles between brain, body, and environment are extended towards the world by the habitual functioning of the lived body, mirrored on the biological level by the structural coupling of organism and environment through circular perception action loops. Continuous incorporation of experience into body memory induces neuroplastic changes in the matrix and synaptic structure of neural networks. Connectivity between brain regions results in more adaptive patterns of neural activity. And over time, experience, habits, skills, interactive schemes, and dispositions that characterize an individual is sedimented in body memory. Interactive experience becomes an organic disposition, which enables new forms of experience as life processes lead to structural change. The construct Psychoneurointracrinology in my book situates the paradigm of neurophenomenology at three levels: psychological, neurological, and intracrinological, forming a mind-brain continuum within the person. Psychological refers to constructs variously referred to as psyche, self, soul, mind, and consciousness. Neurological to the composition and reactions within the nervous system, and intracrinological to the intracellular biosynthesis of steroids, the binding of receptors, and the formation of enzymes that catalyze the creation of hormones within the cell. This model integrates conscious and unconscious processes with the emotions to reveal the person's perceiver-dependent embodied sense of self and well-being. I argue that the sense of self has neural correlates at the interface between the hypothalamic pituitary gonadal and adrenal axes, which are responsible for an active engagement in the development of meaning through their connections to higher order functions of the brain that modulate the reactive and anticipatory response to stress. This co-regulation may occur through top-down activation and bottom-up feedback as coupling of the HPA and HPG axes through parallel involvement of social and emotional relevant circuitry. It is hypothesized that progesterone and DHEA levels fluctuate with the sense of self as the boundaries of the personality are integrated, and cortical limbic projections from the paraventricular nucleus to the median eminence converging with gonadal steroids in the medial preoptic area are an interface between the mind and the brain. I will briefly explore this thesis. This is the table of contents for my book. Each chapter is presented in such a way that readers from diverse backgrounds will follow the progression of ideas. The example of a woman's biochemistry is given. In overview, menarche at puberty derives from the complex interplay between hormones in peripheral circulation, neuropeptides, neurosteroids, and excitatory and inhibitory neurotransmitters inflecting sympathetic and parasympathetic arousal. Concomitant changes in mood, affect, and behavior are associated with the neuroendocrine regulation of the HPO-HP axes and cortical subcortical control of the hypothalamic circuitry. The ovary has a cyclic function under the feedback control of the HPO and HP axes. The HPO axis governs development, reproduction, immunity, and aging. The HPA regulates the fight or flight response to stress and homeostasis. HPO axis governs development, reproduction, and immunity and aging, whereas the HPA regulates the fight or flight response to stress and homeostasis. During the reproductive years, the functional balance between the HPO-HPA axes depend on a woman's need for hormonal substrate to maintain the menstrual cycle and response to stress. This graphic shows the mechanism of the HPO axis. GnRH signals the ovary to secrete estradiol and progesterone, which stimulates the anterior pituitary to secrete FSH, followed by a pre-ovulatory LH surge, ovulation, luteal phase, and menstruation. This graphic shows hormonal change in peripheral circulation measured in plasma and serum during the normal 28-day menstrual cycle. The next slide explains this graph and what happens during stress. Please review this slide later. This graphic shows the rise and fall of neurotransmitters throughout the cycle. Again, see the next slide for a description of this mechanism. Please review this slide later. The HPA axis is responsible for the biochemical reactions associated with the anticipation and perception of stress. Again, see the next slide for a description of mechanism. Please review this slide later. This graphic depicts a holistic autopoietic representation of steroid biosynthesis. Follow along with these steps. At the top of the chart are hormones synthesized from LDL cholesterol and pregnenolone. The same biochemical pathways are present in the ovary, testes, adrenal gland, and the brain via the same catalyzing enzymes printed at the bottom of the chart. See the following two slides for more description. Again, review these slides later. My contention is that intracrine biosynthesis is autopoietic in the sense that the person's perception of experience determines receptor sensitivity in response to local physiological need in both immediate stressful situations and to fine-tune hormone receptor dependent processes. This graphic shows 5 limbic system regions known to affect HPA axis activation. This graphic shows how the paraventricular nucleus receives sympathetic input from CNS neurons, evoking rapid reflexive activation of the HPA axis. This graphic illustrates the central mechanisms of stress integration through projection to the medial PVN from 14 extra-hypothalamic pathways regulating homeostatic functions. Psychoneuroendocrinology is a complex systems model with explanatory value, providing a way to bridge the gap between the mind and the brain, not only how the mind develops meaning and the sense of well-being, but ultimately how the notion of self emerges from this complex system. Please review this slide later. Thank you. Please review the slides I provided. I look forward to your comments and questions. Here is my contact information.